Hello, hi, good afternoon, Shweta. Are you there?
Good afternoon, Naman here, and I'm very sorry that I cannot talk, that I'm traveling right now.
No
Coming to me.
problem. No
I just
problem.
missed the class
No
because I
problem.
cannot afford to miss another class.
Okay, no problem. You can drop message in the chat box. No problem with
Yes,
it.
ma'am. Yes, ma'am. Yes, ma'am.
Okay. So, hello everyone. My name is Kajal Kachawa. I'm PMR Research Scholar from IIT BHU Varanasi. I'm your teaching assistant for this course of Host-Pathogen Interaction, that is Immunology, and the duration for which is January to April 2025. And today we are at week 10 of this particular course series, and the lecture material till the week 9 from my side is already available on my YouTube channel along with the assignments. So you can check it out there if you have not visited yet. And my class timing is from every Saturday evening 3 to 5 p.m. Till now, the lecture material from Professor Himanshu Kumar sir is released on the NPTEL, that is until the week 11 assignment and the lecture material. So only 1 week is remaining, that is week 12. So almost every material is available on the NPTEL channel, so you can check it out there. And today we are at week 9 assignment. So in the last class, I have discussed about the viral pathogenesis. We have started a new topic other than the immune cells and everything. The week 9 is about viral pathogenesis. So there are different patterns of an how virus can infect the host cell and what is their response. So I would like to quickly summarize this. If you have not visited the last week YouTube video, so you can check it out in detail there. But I will quickly summarize, then we'll start the week 9 material. So the first type is acute infection. In this type, it is a rapid onset and it's for very short duration, like as the name suggests, acute infection. So it is for a very short duration and it have a very rapid onset. And this virus is cleared by the immune system very quickly. And the second type is latent infection. This virus lies in a dormant stage after the initial infection and it can reactivate multiple times. Like for after first infection, it can go into the dormant stage and it will reactivate again and again. So this is the latent infection. The third one is progressive infection. One thing it got stuck again. Shweta, can you see the slides movement or not? Can you write in the chat box, please? I think it's stuck.
Yes, ma'am. It's visible. The markings are visible.
But is it moving right now, slides? It's stuck.
No, ma'am. Stuck in the viral pathogenesis classroom.
Okay, just give me 2 minutes to correct this. So sorry, I will correct this and I'll be back to you. Please correct. I'm so sorry. Yeah, I'm so sorry for the technical error. So we were discussing about the acute infection, which have a very short duration, and the latent infection, which can reactivate again and again after a single infection. It lies in the dormant state in our host system. And the third one is the progressive infection, which is after an initial infection, the virus load slowly increases over time, as you can see it here. And eventually it leads to very severe disease or any like severity like immune system failure also. So this is a progressive infection. And the fourth one is chronic or the persistent infection. In this case, the virus is never fully cleared. It always remains there and somewhat extend. So it's like continuous low level of replication is there in this case. And the last one is failure to clear all evidences of infection. In this case, what happens is like this acute infection resolves, but the virus nucleic acid persists there. So there will be a slight amount always there. So these were the five type of viral pathogenesis patterns that we have discussed in our last class. So today we will start the innate immunity during any viral infection, like what how our innate immune system will respond to the viral infection. Till now, we have discussed in general what is innate immunity and adaptive immunity and how they respond to our body, what is their function, what is their role, what are their components. Till now, in our previous classes, we have discussed about that part of innate and adaptive immunity. And today, we will discuss particularly the innate immunity during the viral infection. So in innate immune response, what happens like we have, if you remember, then we have discussed about the PRR and the PEM. These PRR were the pattern recognition receptors and this PAM were the pathogen associated molecular patterns. Like for a short quick revision, this PRR were pattern recognition receptor and the PAM were the pathogen associated molecular patterns. Till now, we have discussed what were these, like these PRR were present on our immune cells, our body's immune cells. These were the receptor for the detection, and the spam were present on the pathogen, on the surface of pathogen, which were being recognized by our immune cells. So there was interaction between this PRR and the PAM. So this is exactly what is happening here. In the case of viral infection, the PAM molecules are the some receptors or some patterns which are present on the surface of the virus, which will be recognized by our immune cells. So there is a case like this response to virus infection. What will happen here? Okay, missed this slide. No problem, we'll cover this here. Like this first one is like direct detection of virus by the PRRs. So what will happen in this case? The viral signatures or the spam molecules, that is the molecular patterns present on the viral surface. These are the viral neuramic acids because viruses are replicating in host cells and they contain the long double-stranded RNA which contain this 5' triphosphate and unmethylated CpG motif in the viral genome, viral DNA genome. So, how this is different from our host cell? There is a point like this. How this viral genome is different from our host cell? What is difference between these? So what happens here when this virus infect the host cells, then they begin to replicate. And during this process, they generate the viral nucleic acids that act as the PAM. Like this is the viral nucleic acid because these viruses will be replicating inside the host cells. So they have the long double-stranded RNA, which is normally not present in the healthy host because these viruses have their double-stranded RNA with 5' triphosphate. 5' triphosphate, and which is not present in our human host cells. And also they have like this unmethylated CpG motifs, which are normally methylated in humans. Like first thing, they have 5' triphosphate, which is not in humans, and they have this unmethylated CpG motif. In humans, in our human, this is methylated. So there is a difference between these both the genomic content of virus and the host. So that's why they are different. These viral signatures are different and they will be recognized by our PRR. And in addition to that, they also, these factors help determine the viral origin. How? Like now, this viral nucleic acid in the endolysis is like this virus, but not the host nucleic acid are normally found in the endolysosomes. And virus, but not the host DNA is present in the cytosol. So there is a difference where this genomic content is present in the case of virus and the host. Their presence and their origin is different between both. So our immune cells will recognize them that this is not our host genomic RNA or DNA, this is virus. And this was the direct detection of virus by PRR. And the second case is indirect detection of viruses by the PRR. So in this case, what will happen? The alteration in normal cellular process, like this acute decline in the host protein synthesis will happen, and the altered activity of the ion channels will happen, or the ER stress will be there. So this is the case where we can see that yes, our human body is getting infected by the virus. Because of this virus infection, these things will happen. Like if a virus is trying to hijack the human cells, then we can see a drop in the host protein synthesis and the ER stress we can observe, and also the ion channels will be altered. So these are the signatures of viral entry in our body. So these are the two ways how we, like our PRRs can detect the presence of PAM. So the first, this was the innate immune response. And in the second case, the response to viral infection, how now our body knows that there is a virus infection in our body. So how they will respond to this. So the first case is this type 1 and type 3 interferon will come into play here. Like these are the signaling proteins which are released by the infected cells. And like they are the emergency broadcasters type of, like they will alert their neighboring cells that there is some virus infection in our body, so get activate. Like other immune cells, they will get activated. So when a human cell is infected by virus, they will release this type 1 and type 3 interferons and alert the neighboring cells. And the second is the pro-inflammatory cytokines. Like these molecules will help to recruit the immune cells. Like we have discussed the macrophages and dendritic cells. Then they will come to the site of infection. Then they will increase the blood flow there, and they will increase the blood vessel permeability, and they will help the immune cells to move quickly to the affected tissue. So what will happen if there is any virus infection in our body. If any virus is infecting any cell, then the cell will release the type 1. This will release the type 1 and type 3 interferon to the neighboring cells. And these cells will now they will release the this pro-inflammatory cytokines, and they will call the macrophages and dendritic cells to the site of infection. Then what they will do, they will increase the like this membrane permeability and the blood flow. Then ultimately other immune cells will reach there. So this is the innate antiviral mechanism. But what will happen in the case of this adaptive immune system antiviral mechanism? In this case, the like as we know that our Adaptive immune system is long lasting and it is very specific, and it is capable of developing the memory what will happen. Yeah, so in the case of adaptive immune system response to virus infection, what will happen? They are the antigen specific and have the long lasting antiviral immunity, and they will call upon or they will take help of this CD8 T cells and the NK cells. These will release the perforin and granzymes to the infected area, and they will kill the cells, and this NK cell will target the infected cell, and like they will start the MHC1 expression, and that everything you have already studied in the previous classes. And they will what do they will form the neutralizing antibodies, and these antibodies will they will bind to the viral particles and block their entry into the host cells. So these antibodies will block the viral entry to other host cells, and they will block their uh, protein synthesis mechanism and their replication. So this is the case with the adaptive immune system, how that will respond to this. And all these mechanisms ensure inhibition of viral replication and viral genome expression and virion assembly. So overall, the role of this innate viral antiviral mechanism or the adaptive antiviral mechanism is to inhibit the viral replication and viral gene expression and the virion assembly. Like they have to inhibit this. This is their ultimate aim. So this is the response to viral infection from both the innate and adaptive immunity, which is being discussed in detail in the upcoming slides. So the first we start with the innate immune response against viruses. So what will happen in the case of innate immune system? We have seen about type 1 interferon and this type 3 interferons, and also the type 1 interferon inducible genes and the role of inflammatory cytokines. So this type 1 interferon, if we discuss about, then like this consists of this, like in type 1 interferon, interferon it includes interferon alpha and interferon beta. And this interferon alpha is 13 member and this interferon beta is single member. So there are two subtypes of this interferon 1. And these interferons are produced very early after the viral infection by the dendritic cells and the macrophages, and their primary role is to induce the antiviral state to the neighboring cells, like to allow or the like to alarm the neighboring cells that yes, there is an virus infection in our body. So this is their role, primary role. And this, when we talk about this interferon 1 inducible genes, then these genes are they activate a family of downstream genes known as the this interferon stimulated genes. Interferon stimulated genes. So when this type 1 interferon is produced, they will produce their downstream genes, which are interferon stimulated genes or ISGs, and their example is this IP10 and this rentase. That is their example. So what is this IP10? This IP10 is a chemokine that attract the immune cells like T cells or the NK cells to the site of infection. The role of IP10 is to attract this T cell or NK cells, like it is chemoattractant, and the role of this rentase is to recruit the T cell or the eosinophil and basophil to the site of infection. So what is happening in this case here? These inducible genes, there are two types. The example is two type, IP10 and rentase. This IP10 will, they will call upon the or attract this T cells and the NK cells, and this rentase will recruit this eosinophil and basophil to the site of infection. And the third mechanism is by this interferon type 3 interferon, and it consists of it have like three members in its family. Like this interferon 1 had two members that were interferon alpha and interferon beta, and this interferon type 3 have three members in its family. So its basic role is to give a localized effect, like to act on a local site. Like interferon 1 was calling upon the cells there, and interferon 3 will act on that site. And the last one is their role is inflammatory cytokines. Like we have already seen some interleukins and some tumor necrosis factors. So this will also come to the site of action. So these are the factors involved in the innate immune response against this viral infection. So this is the mechanism of this virus infection. What is happening when a RNA virus enters into the cell, then its viral RNA is being recognized by this RIG1, and some factors are forming, and this inflammatory cytokines and interferons will form the they will cause the antiviral response, and this then the degradation will happen of the viral genome, and they will not be replicated in the upcoming cells or in the nearby cells. So this is a pathway you can see in the detail from the video. And this is your example of this influenza E virus infection. What will happen in this case? Again, the inflammatory cytokines and the interferons will be released, and they will form the antiviral response, and this viral replication will be inhibited or stopped there. So these are the pathways you can see there. And this topic is interferon type 1 interferon independent antiviral defense. Till now we were seeing interferon dependent antiviral response from the innate immune system, and here this is the type 1 like interferon independent antiviral response. So what happened in this case is the clearance of intracellular pathogens via autophagy, which is followed by degradation of the lysosome, which is referred to as xenophagy, and it presents an important mechanism of antiviral defense. So, what is happening in this case here? Let's see on this slide. So, first of all, let's see what is this xenophagy. This is a type of autophagy. If you have heard about autophagy somewhere, then you might be knowing about this, that degrading or like eating up something. So this genophagy means it is a cellular process of degrading and recycling the cytoplasmic content. So what happens here? The degradation and recycle of cytoplasmic content, and in this genophagy, and this process is redirected to target and eliminate the intracellular pathogens. So let's see step by step what is happening here. The first one is like recognition and the engulfment. What happens here when a virus enters into the host cell? It is either a whole particle or any intermediate of the virus cell, so it can be marked for the degradation. This is the host cell here, and this virus is trying to enter inside this cell. So what will happen is here around when this enters inside the cell, then a phagophore is trying to form around this virus cell, like a folded membrane type of structure is trying to form around the virus cell, and then it will form the autophagosome. This is like phagophore which closes to form a double membrane vesicle, which is known as the this autophagosome. This is double membrane vesicle, you can see it here, which will trap the virus inside them. Then it will go and fuse with the lysosome, and as you know that this lysosome contain the digestive enzymes, and this will form the phagolysosome. Then what will happen? This this fused phagosome and lysosome, it will get degraded because of the lysosome enzymes, because of the release of the lysosome enzyme and this viral particle, it will get degraded here. So it will not replicate inside the cell. So this is a mechanism of where here we can see that no interferon is involved in this process of the xenophagy. Only the this lysosome mediated killing of the virus is there. So this is the virus evasion of the innate immune system. What will happen? Like all the pathogen viruses, they encode factors to counteract the induction, signaling, and antipathogen effector functions. So what happened in this case is every virus, whether it is influenza, HIV, or any herpes virus or any virus, they will encode for the immune evasion proteins. They will encode for the immune evasion proteins. These viral proteins, they will disrupt the different stages of the interferon mediated antiviral response. Like this virus have their own defense kind of protein, so that they want to disrupt the interferon, and they want to try to cause the infection there. So this virus employ distinct molecules to achieve blockage of the antiviral effects of type 1 interferon, and these can be categorized into four major categories. The first one is global inhibition of cellular gene expression, which is what happens here. This virus can shut down the host transcription and translation machinery and make it impossible for the host to produce any type of antiviral protein. Like it will globally inhibit the cellular machinery of the host, so that host cannot make any antiviral protein and cannot stop the viral infection though. So first mechanism is that. The second one is evasion from innate recognition. In this case, the virus will hide their RNA or DNA from the PRRs, and they will try to evade from the innate recognition, so that they will be not recognized by the PRRs. And the third one is inhibition of molecules involved in the interferon induction and signaling. So in this case, even if a virus is detected, it can block the signaling pathway which is needed to produce the interferons, so that they will not be produced. It will inhibit or interfere between the interferon production pathways. The last type is inactivation of the interferon induced effector molecules. In this case, what happens? These interferons stimulate like hundreds of antiviral proteins, and this virus evolved the mechanism to neutralize or degrade these effectors. Like even if the pathway, like here, first of all, what they were trying to do, they were trying to stop the host gene expression machinery. But by chance, if they were unable to stop this machinery, then what they were trying to do? They were trying to hide their own RNA or DNA, so that it can't be recognized. And if they were unable to hide their genomic content, then what they were trying to do? They were trying to interfere with the interferon production. Interferon. interfere with the interferon production pathway so that interferon will not be produced and they will not be recognized. And even if they cannot stop these pathways or cannot interfere with these pathways, then their last option was to evolve some mechanisms so that sorry, evolve some mechanism so that they can neutralize or degrade these interferons. So these were the four types how they are trying to block the antiviral effects of interferon, or they want to hide from the innate immune system. So these were their mechanisms, and they also have immune evasion proteins in them. So this is a mechanism of innate immune system, how virus is trying to hide from the immune system, or how the innate immune system tries to kill the virus. So this was the overall about innate immunity. The next, I hope this is clear till here and no doubt is there. If any, then you can ask or discuss, or you can drop a message in the chat box. I will take from there. The innate immunity part is over here. So I hope this is clear. Okay. Then let's move to the adaptive immune system. If you have any doubt, then you can post in the chat box. Now let's move to adaptive immunity during viral infection. Okay. So till now we have seen the role of immune cells and the interferons in the virus infection. Now we will see the role of antibodies involved here, because now we are talking about adaptive immune system. So, like these antibodies don't just only neutralize the virus before the infection, but they also act against the infected cells. Like, first of all, they will try to neutralize the virus on their entry, but if not, then they also act against the infected cells. So, there we'll see there are different steps of how an antibody is acting against the virus infected cell. So, like they will neutralize antibodies against free virus particles. Like they will, this neutralizing antibodies will bind directly to the virus surface protein. These neutralizing antibodies will bind to the virus surface protein and they will neutralize them. Or they will have a complement mediated lysis or the phagocytosis. So then the cell will be protected from the infection. This is their first way of stopping the infection. And if the antibody is unable to block this, then what will happen? They will act on the infected cells. So in this case, what they can do, these antibodies have the Fc mediated effector system. If you remember the structure of antibody that we have already discussed in the previous week classes, then there were two regions. First was this FAB on the upper side, and here lower side it was the FC. The upper one was the fragment of antigen binding, and the lower one was the fragment of crystallization. So this antibody from here it will bind to the antigen, and from here this lower portion it will interact with the immune cells or the body cells. It will bind to those cells here from the FC region. So this sorry. So these antibodies have the Fc mediated effector system in its the Fc region, and these antibodies will bind to the viral protein expressed on infected cell surface, and they can engage the immune cells so that they will infected cell will get lysed or they will be cleared off by the immune cells. And the second is their inhibition of virus replication or the cell signaling. Like some antibodies will affect the intracellular signaling of the virus and they will not allow them to replicate. So they will inhibit the virus replication. And in the third case, what they will do, they will inhibit the virus release and the transmission. Like they will cause the steric obstruction and inhibit of virus release. Like if they are unable to interfere with the virus replication machinery and if the virus have already replicated inside the infected cell, then they will interfere with the like the virus have to release out from the infected cells so that they can infect the nearby cells. So this what the antibody will do. They will interfere with the release and the transmission to nearby cells so that they can stop the infection to that particular cell only. And in the last class, what they like they will have this steric obstruction here so that they will also stop the cell to cell interaction or the cell to cell transmission so that this virus will not infect the nearby cells. So these are the mechanisms of how an antibody will interact with the virus. In the first part, either they can interact with the sorry, either they can interact with the virus surface protein at outside only and neutralize them and protect the cell from infection, or they can also interact with the infected cell. And there are these four type of ways how they can stop the virus replication, infection, and transmission to other cells. So I hope this part is clear of how antibodies interacting with the virus infected cells. And there are some this effector activation of activated CD8 and CD4 positive cells, like we have seen in some previous classes about MHC class 1 and MHC class 2, major histocompatibility complex MHC1 and MHC2. So I hope if you remember from there, then we have studied that this MHC class 1, this will interact with the CD8 positive T cell, and this MHC class 2, this will interact or present antigen to the CD4 positive cell. So there were two classes of MHC in the previous classes, if you remember correctly. Then there is this effector type. This is CD8 positive cell or CD4 positive cells, and they will interact with the target cells, whether MHC1 or MHC2, and their effector mechanism is like either there can be direct cytolysis or any release of the granules like perforin and granzymes, and there can also be some other cytokine synthesis and other mechanisms also of like if a virus is presented to an immune cells, then what will be their effector mechanism? It depends on what type of effector type cell is presenting to them. So there are like synthesis of different type of the cytokines and like interferon, interleukin, chemokines, and release of granzyme perforins. So there are different types of how a cell will interact and how a virus will be stopped from the replication. So the next part is adaptive immunity, adaptive immune memory. Like we know that adaptive immune system have a memory stored with them. So, like this memory, B and T cells will be generated after an infection or the vaccination, and these are generally long lived. Like they will be stored in our body, and the factors controlling the formation or the duration of the memory immune responses are not yet well understood, but several cytokines like this BAF, that is B cell activating factor, these may play an important role in maintaining the viability and the basal proliferation of the memory cells. Like there will be B cell memory and this long term humoral immunity. So, what happened in this case is here, like this B cell activating factor, which is BAF, it is a key survival factor for B cells, and it helps memory B cells to maintain to, or to remain viable and to continuously proliferate even in the absence of this memory cells. So this is a very important factor which plays a role in the adaptive immune system to maintain its memory. These are the BAF. And like there are two principal mechanisms. Like first one is repeated exposure to antigen is one way in which memory B cell level can be maintained. Like our body should get repeatedly exposed to the same antigen so that our memory cells can be formed continuously. But all the time this is not possible. So what will happen? The pre-existing serum and the tissue high affinity or the class switched antibody to virus probably form the long-lived plasma cells in the bone marrow so that they will be continuously formed. Like even without the re-exposure, some plasma cells will be derived from the memory B cells and they will migrate to the bone marrow. And these cells will secrete the high affinity or the class switched antibodies for months or years so that that particular antibody will be continuously present in our body for any future exposure to the same antigen. So these are the two types how we can have continuous memory cells present or formed in our body. And next part is like this long-lived plasma cells. They secrete this high affinity antibody, which is long-lived, and they survive without cell division for many years in human and contribute to the maintenance of antibody levels over decade. So whatever role is to here, if we got infected from any particular virus, then our aim or like our interest is to maintain the antibodies against that particular virus or antigen so that we have the memory cells continuously in our body. So these are the different ways how we can have this memory cells in our body. And the next one is this antigen independent or the bystander activation. So what happen in this case is the plasma cells can be maintained without the re-exposure to antigen. And this is referred to as the bystander activation. This helps to know that why this antibody titer can remain stable for decades after the vaccination. Like there are continuous without the exposure or presence of antigen, the antibodies continuously found in our body so that we have a long term memory for that. And there are some short-lived plasma cells also, like they survive for only few days and they produce antibody in extrafollicular foci. And they are probably crucial in very early response to pathogen. So in this case, like these cells are generated rapidly during the infection or after an vaccination, and they reside in the peripheral tissue and particularly in the extrafollicular foci of the lymphoid organs. If you remember the structure of the lymphoid organ, there were follicular region and other region. So these cells remain present there for a time, but their lifespan is very short. So they are critical for the initial surge of the antibody production. Like there are some studies on how they have studied about different viruses, like their how they are like how antibody titer is maintained over the decade or that will last in shorter time. So there are some studies, you can check it out from the lecture videos in detail. Then there is this T cell memory. I hope you remember about the T cell and the B cell that we have already studied. Like there are some effector T cells and there are some memory T cells. These effector T cells are higher frequency and their lower activation threshold to trigger cell proliferation and differentiation. I hope these things are already taken in the B cell and T cell lecture videos of about like there are two type of cells. First one is effector, which will act at that time, and the other one is memory cell, which will be stored for any further infection in the future. And there are role of memory CD8 positive T cells in the virus infection. There are some viral strategies to evade the adaptive immune response, like they can escape by mutation, or they can escape by the latency, or they can escape by the destruction of the immune cells. There are different ways how this virus can uh, evade from the adaptive immune system. Like they can get mutated so that they will not be recognized by the immune cells, or they can be escaped by the latency, like they can enter in the latent state so that they will remain hidden from the host immune system for a longer time. Then after some time they will start expression and infect the host cells, and they can also escape by destruction of the immune cells. Like there are some cells, like in the case of HIV, that that can kill the CD4 positive T cells, and that is also the case. Like this virus can also kill the immune cells, and they can also escape by subverting antigen processing and presentation. Like they can block the antigen presenting cells, like they can block this MHC. So virus will block this MHC so that there will be no antigen presentation and processing. And they can also inhibit the T cell mediated target cell lysis. Like they can block the release of this perforin and granzyme action so that they will not be killed. And there are the inhibition of inflammatory response via modulation of cytokine action. Like they can also block the cytokines, and like there were in the previous slides we were seeing about how it can block the interferon production, and they can also inhibit the humoral immunity by virally encoded Fc receptor, complement receptor, or the control protein. Like they can block the humoral immunity and the receptors. So there are different strategies how a virus can evade the adaptive immune response. And the inhibition of class 1 and class 2 MHC complex synthesis also can be inhibited by this virus proteins, which are known or already reported to suppress or inhibit the MHC gene promoter activity. And there are this HCMV, that is human cytomegalovirus. It can impair the MHC class 2 expression, so that if the MHC or the like antigen presentation is not there, then immune cells cannot recognize virus entry. So virus can freely replicate in the host cell. So there is also inhibition of MHC class 1 and class 2. And this is the viral evasion of humoral immunity. Like there we have seen the evasion of the innate immunity. Here, this is the virus strategy to evade the neutralizing antibody response to the virus surface protein, which is like they can mutate their surface protein by the antigenic drift and the antigenic shift, and also by the recombination. And this most notably for highly variable viruses such as the influenza virus, HIV, or HCV. These are observed in the case of these particular viruses. Like they can mutate their surface protein by the antigenic shift and the antigenic drift that we'll discuss in the coming slides. And they can also mask their conserved epitopes. Like these epitopes are the parts of the like antigens which are being recognized by the antibody. So they can, there are some conserved regions in the epitopes, so they can mask that, they can hide that so that they will not get recognized by the antibodies or the adaptive immunity. There is also a mechanism of glycan shielding as seen for HIV and the Ebola viruses. Like this can also happen in some viruses like HIV and Ebola, where what will happen, like they can add the carbohydrate. These glycans are the carbohydrate groups. So these viruses can add these glycans to their envelope protein, and these glycans are poorly immunogenic. Like they create a sugar coat that blocks the antibody access. So they can form a shield of the glycan around their antigens. Like they can form a shield of carbohydrate sugars present around them so that they will not get recognized by the immune system. And there are also mechanism of surface protein decoys that may move up neutralizing antibodies under certain conditions. This happen in the case of this HBV. Like what will happen in this case, they can release the soluble virus proteins into the circulation, and this decoy protein will bind up with the neutralizing antibody and they will prevent them from reaching the actual virus. So they will release the soluble viral proteins so that, like if our body is forming 100 neutralizing antibody at a particular site, then these surface protein decoy will release 100 this fake soluble viral proteins, and that antibodies will get neutralized and there will be no antibodies present to bind with the actual antigen. So this is also mechanism, like these viruses are so smart, and they can additional antigenic sin, which will happen in the case of HIV or the influenza virus. What happened in this case, like the immune system prefers to recall the memory B cell against the initial infection of any virus, and this limit the generation of the new effective antibody response to mutated version of virus. Like there will be original antigenic sin so that this new antigens cannot be recognized by the antibodies. And they can also, like there is irregular surface protein spacing that tends to reduce immunogenicity. Like they can also change their surface proteins. And the multiple surface protein and virus forms, for example, in the case of pox virus, which encode multiple proteins for entry. Like our immune system can form antibodies towards two or three surface proteins at a time, then this virus will encode a new surface protein so that it can get entry into the cell. So there are different mechanisms of how this virus want to evade from the adaptive immune system. So these are few of them. And there is a virus induced immune dysregulation and also the autoimmune disease is there. Like in the case of virus infection, particularly the chronic viruses, which are shown to enhance the autoimmune disease in the susceptible individuals, as infections frequently induce strong inflammatory response in the various organs. So they can also induce the autoimmunity in the host body. And there are several major pathways through which viruses can initiate or more likely modulate the autoimmunity. The first one is direct infection of target cell or organ, which can cause the release of sequestered autoantigens and enhance the antigen presentation. So what is happening in this case? Like this direct infection of tissue, uh, which can cause damage or the cell death. This leads to the release of self antigens that are normally hidden in our body, and like they are known to seen as foreign, and they can be presented by the antigen presenting cells, which will activate the self reactive T or B cells. So like there are different ways of like this how local inflammation might alter the repertoire of self epitopes presented by the antigen presenting cells by altering the antigen degradation properties of the proteasome. And they can also the presentation of pathogen epitopes with structural or sequential similarity to self epitopes, which might specifically activate the autoreactive lymphocytes, or they can cause a molecular mimicry, like they can activate the autoreactive lymphocytes, which will kill their own lymphocytes. So these are different ways how they can dysregulate our immune system. So there is an example of this influenza virus, which is discussed in the detail. So I hope you have gone through this already from the lecture video, like how this was discovered and what was the story behind Spanish flu pandemic, from where the case of this influenza virus came into account. Yeah, so let's discuss this influenza virus structure. There are different type of these receptors present in the influenza virus by which it can cause infection or it can get entry into the host cell. So let's look into these. There is a heme agglutinin, which is like it attaches virus to the respiratory cell receptors. And there is this neuraminidase, which mediates the cleavage and release of the new virus. And there is here this inside. This is these are some molecules of PA, PB1, PB2. These are heterotrimeric polymerase, which are required for the virus replication and transcription. And there are some nucleus export proteins which regulate the nuclear export, replication, and transcription. There are this BM2 molecules which are integral membrane proteins which are important in virus infectivity. There are this BM1 which is a matrix protein, or this is here a matrix protein inside outside the virus code. And there are some additional influenza B proteins which are required during the infection. The first one is NB, which is responsible for optimal viral RNA localization and encapsulation of viral genomic RNA. There is NS1 molecule, which antagonizes the host interferon response, and there is this NB molecule, which is integral membrane protein. So there are different receptors and different molecules present on a virus surface which are required for its replication and the infection. So there are some proteins which are required for this viral assembly. There are some, this M1 is required for virus assembly, and this M2 is required for virus uncoating, and this Na is required for virus budding off, and this H is required for the attachment of virus to the host surface. And there are some integral frameworks, and there are some inhibitory host machinery proteins like this NS will inhibit the host machinery. So there are some structural and non-structural proteins present there. And there is a polymerase complex which will be helpful in making the copies of the virus, that is for the RNA transcriptase. Yeah, so there are so many proteins and molecules present on the virus surface which is required for us its functioning. There are four types of influenza virus. First one is A type, which infect the humans and has pandemic potential. And the second is B type, which infect human and has the epidemic potential, but cannot infect animal and can cause epidemic. The third one is C type of influenza, which causes the mild illness to human and it lack the this Na. And there is D type which infect the pigs and the cattles and no infection in the human. Like there are these four types of influenza. And there is this nomenclature of this viruses, like how they are being given names. Like we know there are in the name of virus there are some symbols, letters, and there are some cities sequence. So how they are named, there is a nomenclature of this virus. It can be on the basis of this antigenic type, that it can be A, B, C, D in the alphabetic order. There can be the host origin from where it is being originated. It can be on the basis of geographical origin from the which place it is first being identified, and it can be strain number, it can be year of the collection. There are these different type or the criteria for the nomenclature of this virus. So there are two main receptors in the this influenza virus. First one is the hemagglutinin and the second one is the raminic acid receptor. So what is their role? This hemagglutinin is for receptor binding and the fusion function. The major function of this HA are the receptor binding and the fusion activities, but there may also be structural role of the HA in budding and the particle formation. And this HA is a trimeric rod shaped molecule, whereas the Na or the nuraminic acid is tetrameric, and this HA have a carboxylic terminus inserted into the viral membrane, and it also have the hydrophilic end projecting as a spike away from the viral surface. And the overall amino acid sequence identity can be less than 50%. And the structure and function of these HAs are highly conserved. So these are the receptors on the virus surface which are required for binding and the fusion to the host cell. And this how influenza virus infect the humans. That is, there is a pathway of I told you about the antigenic shift and the drift. So there are the antigenic determinants. So we'll discuss about this. Now let's start the questions of the week 9, that is assignment 9. So the due date is already over on the 26th of March. I hope you have submitted this. So I hope till here the lecture topic is clear to you. If not, then you can ask the question if you have any doubt. We will discuss that. Otherwise, we will move in solving the questions. Okay, no doubt is there. If you have, then you can let me know. So the first question was which characteristic distinguishes viral nucleic acid from the host nucleic acid, facilitating PRR recognition. This we have seen that in the virus there is a presence of the long double-stranded RNA structure in the cytosol. Like we have seen in the case here, there were this virus contain a long double-stranded RNA which contain the 5' triphosphate and unmethylated CpG motif. And also the presence was different that it is present in the cytosol or where it is present. So in the case of this virus RNA or the virus nucleic acid, this is different. So we can differentiate it from the host nucleic acid because of its presence in the cytosol and also Uh, like the next option is this high level of 5' triphosphatase on RNA molecule. No, it was 5', not 3'. And the highly methylated CpG motif, no, in virus it was unmethylated CpG motif. And the exclusively nuclear localization of viral nucleic acid, no, it was present in the either in the cytosol or the endolysosome. And the frequent presence of polyadenylated tail on the viral RNA. No, this tail is present on the host nucleic acid, not on the virus. So the correct answer to this question is presence of long double-stranded RNA structure in the cytosol. The next question was which mechanisms are known to be triggered by the type 1 and type 3 interferon as part of the antiviral response. Like they are talking here about type 1 and type 3 interferon. Sorry. About this type 1 and type 3 in the antiviral response. This we have already seen in detail about this type 1 and type 3. Like they induce the apoptosis in the infected cells and they activate the this interferon stimulated genes or the inducing genes, and they down regulate the virus replication pathway, and they also recruitment of professional phagocytes, that is the macrophages and dendritic cells. So these are the correct answers to the MHC type 1 and type 3 interferons. They will not directly neutralize the viral particle. They will call upon some immune cells which will bind with those receptors and they will kill the virus. So they will not directly neutralize. And the enhancement of host cell protein synthesis, no, it will not get enhanced. And activation of antigenic specific cytotoxic T cells, no, this will happen in the case of adaptive immune system where the CD8 positive cells will get activated. And the stimulation of B cell proliferation, no, B cell will not be involved here in the case of interferons. So the correct Answers are induction of apoptosis in infected cells, activation of ISTs, and downregulation of virus replication pathway, and recruitment of professional phagocytes. The third question was, which of the following mechanisms involve evading neutralizing antibodies while altering viral surface proteins by highly variable viruses like influenza virus or HCV? That is the antigenic shift and the drift. Just a minute. Yeah, so this is the which of the following mechanisms involve evading neutralizing antibodies by altering the virus surface proteins by highly variable viruses like influenza virus and the HCV. This we have seen in the previous slides that in the case of this influenza virus or HCV, what was happening here? The influenza virus and the HCV, like the mutation of surface protein by antigenic shift and the drift and recombination was happening, and it was getting evaded by the neutralizing antibodies. So, what is the antigenic shift and the antigenic drift? What happens in this case? There are two different terms. These are the antigenic shift and antigenic drift. So, like this, sorry. In the case of this antigenic shift, what happens? Like this, first we see the antigenic drift. It is a small gradual mutation in the viral genome, and especially in the genes coding for the surface proteins, and it accumulation of mutation over time. There will be small slight changes in the virus surface protein, and this will get altered. Like because of these mutations, there will be change in the antigenic epitopes. So this is the antigenic drift. It is a small change, and it is a gradual. Over time, it will get mutated and it will change the surface proteins. Whereas in the case of antigenic shift, just a minute. Yeah, so in the case of this antigenic shift, what happened? This is a major or an abrupt change that was small and gradual. It is a major abrupt change. What happened in this case? Like this two different type of this viruses, they will merge together and form a new type of virus. Like their nucleic content will be changed, their surface proteins will be changed, their coating will be changed, everything will be changed, and combining of RNA viruses. Two different viruses will combine and form a third different virus that will be completely different. So it is a major, and this will happen suddenly. So like you can see here, in the case of this antigenic drift, this is a gradual alteration in the viral genome, which is caused by mistakes in the replication or by the mutations, which are random changes. And in the end, what will happen? It will create a new strain of virus. Only new strain will be formed. In general, the antigenic drift within the genes that will code for the antigenic binding sites, it will reduce the effectiveness of the antibodies which are made in the earlier exposure. And this make the spreading of disease within the host less likely, like this host will not get easily affected. And this antigenic drift causes the loss of the immunity as well as the mismatch of the vaccines. Like this will not act upon or it will not get affected by the previous memory cells formed or the previous antibody formed. So this is antigenic drift. The second one is antigenic shift. In this case, what happened? It is a swift and massive antigenic shift in an infection, and it typically required for two different strains of virus to be combined to create an entirely new subtype of virus. Here it was forming a new strain of virus. Here completely new subtype will be formed. And this new type of virus has a mix of antigens on the surface of the original strains, and this is a type of the reassortment that also results in the phenotypic changes. Like all the genetic material will be reassorted, like two different viruses will form a third virus, so it will be completely different. So there are two different terms, antigenic shift and the antigenic drift. So the question was, in the case of this influenza virus and the HCV, how it is getting evaded from the neutralizing antibody. So the correct answer to this is by antigenic shift and the drift. The next question was, which of the following proteins in the influenza virus genome is primarily involved in the inhibition of type 1 interferon? We have seen in the case of this influenza, what they are asking here involved in the inhibition of type 1 interferon. See here we have discussed about this NS1 gene. It antagonizes host interferon response. Like this will inhibit the synthesis of type 1 interferon. It will interfere with the pathway for type 1 interferon. So this is the NS1 gene. The next question was, which virus is specially recognized by RIG despite being a DNA virus? So the answer to this question is the Epstein-Barr virus. I don't know whether you are aware of this different type of viruses or not. Like every virus have a different type of genetic material and different way of its infection. So here the case of like some viruses are single-stranded RNA, double-stranded RNA, single-stranded DNA, double-stranded DNA. There is different type of the nucleic acid content. So here this Epstein-Barr virus, it is a double-stranded virus. It is double-stranded DNA virus. But then also it is getting recognized by this RIG1. Like see, there are this two type of receptor. One is RIG1, second one is MDA5, and their role is different in recognition. Like this MDA5 is responsible for recognizing the long strand of double-stranded RNA, and this RIG1 is responsible for recognizing the single-stranded RNA. So this Epstein-Barr virus is, even though it is a DNA virus, but still it is getting recognized by this RIG1, despite being DNA virus. So the correct answer to this is Epstein-Barr virus. The next question was, what role do cytotoxic T cells, that is CD8 positive T cells, play in the adaptive immune response? They are talking here about the CD8 positive cells in the adaptive immune response. So, like we have seen in the previous slides, that they induce the apoptosis in infected or abnormal cells through perforin and granzyme release. The other options are like they exclusively produce antibodies against pathogen. No, CD8 positive cells will not produce the antibodies. They rely solely on the antibody mediated neutralization. No. They activate B cells without the need for cytokines. No. They suppress the immune response to prevent autoimmunity. No, they will not play any role there. So the correct answer to this question is they induce the apoptosis in infected or abnormal cells. Like see here, the CD8 positive cells and the target cell is MHC1. Like they will have a granule exocytosis. That is, they will release the perforin and enzymes as their effector mechanism, and they will kill the virus. The next question was, what is the key strategy viruses use to evade the immune system? Very simple, like they suppress or destroy the immune cells to escape the immunity. Like they enhance immune signaling to avoid detection? No, they will suppress the immune signaling. They completely block antigen presentation in all cells? No, this is not possible. They increase antibody production to overwhelm the immune system? No, they suppress or neutralize the antibodies. They prevent cytokine release without affecting other immune functions? No. So the correct answer to this question is they suppress or destroy immune cells to escape from the immunity. The next question was, what key concept does the myxomatosis experiment demonstrate in host pathogen interaction? They are talking here about the myxomatosis experiment in host pathogen interaction. So I hope somewhere in the lecture slides you might have studied about this, but I will quickly summarize it here. What was the myxomatosis experiment? It was experiment for studying the host pathogen co-evaluation. So what was this? There was a virus named Myxoma virus. It was a highly virulent pox virus, and it was introduced in Australia to control the overpopulation of European rabbits because this virus infects the rabbits. So what was its initial outcome? Initial outcome was the virus caused extremely high mortality, that is about 99% in the infected rabbits, and it was like devastatingly lethal. It almost killed all the rabbits there. But over time, what happened? The virus evolved into less virulent strain. And meanwhile, the rabbits also developed some genetic resistance to the virus. So, like both were getting evolved here. The virus also got less virulent, and rabbit also developed the resistance to this virus. So, over time, a balance was formed there, and the less virulent viruses that did not kill host too quickly were more likely to spread, and the highly virulent strain killed their host too rapidly, reducing opportunities for transmission. So that there was no transmission of this virus from one rabbit to another rabbit, because the less virulent viruses that did not kill their host and the highly virulent strains killed their host very rapidly, so that they don't have time to get spreaded from one host to other. So overall the spread was reduced in that particular area because the virus also got evolved and the rabbit also developed genetic resistance. So it was a case of this co-evolution here. So this is what they are asking about here. The key concept in the myxomatosis experiment demonstrated host pathogen interaction was that the pathogens often become less virulent over time to maintain survival within the host population. So this is the correct answer to this experiment. The other options are host always evolve to completely and eliminate the pathogen. No complete elimination is not possible, and the pathogens increase in virulence over time. No, it decreased in virulence over time, and the host became genetically identical to resist infection. No, and the virus can no longer mutate once inside the host. No, it became less virulent over time, but mutation was not there. The next question was, what role do small interfering RNAs or siRNAs play in antiviral defense? So, what happens in the case of this single small interfering RNA in virus defense is that, like, they will recruit the RNA induced silencing complex, that is RISC, to cleave the viral genome and provide the sequence specific defense there. The last question was, which of the following are viral strategies to evade the host innate immune response? This is the overall summary or the story of like how a virus strategize to evade the host innate immune response that we have seen in the very previous slides. Like the global inhibition of host gene expression, like it can interfere completely to stop or interfere with the host cell machinery, and it can suppress the type 1 interferon signaling, it can inhibit the PRRs, it can target the immune response stages from detection to transcription, it can suppress the host transcription machinery also. So these are the strategies of a virus to evade from the innate immune response. And the other options are like activation of transcription factor to enhance immune response. It will suppress or inhibit the transcription factors to lessen the immune response. Increased activation of adaptive immunity. No. Deactivation of co-stimulatory signals. So it cannot deactivate. It can just interfere with that. So these were the questions from your last week assignment. Like these were the viral evasion that we have already seen. Until now, the topic covered on the NPTEL channels are these topics. And recently, the bacterial, after the virus infection, the bacterial infection immunology is recently started in the last week. So, in the upcoming classes, we will see that. So, this was from my side till here. If you have any doubt or any question, then you can ask. You can drop a message in the chat box or you can ask.
Not yet so far, ma'am. Thank you very much for understanding my situation also.
It's okay, no problem. Thank you so much for joining and listening to the lecture. You can check out from the YouTube lectures. I hope you have
Yes,
already
ma'am.
visited the last week my video lecture.
Yes, ma'am.
Was that
Uh,
clear to you?
yes, ma'am. It was clear for me. And when I revise, I may get some questions. I'll drop the questions to you.
Yes, sure. You can drop me a message or mail anytime. No,
Yes,
no problem with that. You can revise all the video lectures from my YouTube channel or from also the lecture material from the Himanshu Sir's channel. You can check it out there. If you have any doubt or any question, you can drop a message or mail, or you can also post on the discussion forum if you want. Otherwise, you can drop me a personal mail or message. I will help you out in that. So keep learning and thank you for
yes.
joining.
Thank you so much.
Okay. Thank you. If you want, you can log out.
Yes, ma'am. Thank you very much.
Okay. Thank you.